Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
If you're listening to this, chances are you felt it at some point. That dull ache when you stand up, the stiffness that makes just bending your knee feel like it's not working right.
We're talking about joint pain, and specifically the slow, relentless progression of osteoarthritis.
It is relentless. It's not some rare condition, is it?
Not at all. It affects a staggering 1 in every 5 adults in the United States. It is by far the most common form of arthritis in the world. And for millions, it means chronic pain and a serious loss of mobility.
The scale of this is it's massive, not just in the personal suffering, but financially. What are we talking about in terms of cost?
We're looking at estimates of over $65 billion. That's billion with a B. And that's just in direct healthcare costs each year. It's an enormous economic burden.
And for all that money, what's the current reality for a patient? It's pretty grim.
It is grim because the standard treatments, they're limited. They're almost exclusively focused on managing the symptoms.
So pain control, physical therapy, injections, things to make it more bearable.
Exactly. All just trying to manage the discomfort until the disease progresses so far that the only option left is major surgery, a total knee or a total hip replacement.
And that's the key point. There is no drug out there, nothing approved, that can slow down or, more importantly, reverse the actual damage.
That fundamental thinning of the cartilage has always been seen as a one-way street.
But what if it isn't? That's what we're here to talk about. A new study led by researchers at Stanford Medicine is challenging that entire idea.
And that's our mission for this deep dive. We're going to unpack what they found, and it's a truly revolutionary concept, the potential to regenerate functional joint cartilage.
Not with surgery, but with a highly targeted molecular treatment.
We're looking at sources that detail a protein inhibitor that didn't just stop cartilage loss in old mice, but actively rebuilt tissue. And not just in mice. In human tissue, samples from patients with severe end-stage osteoarthritis.
If this translates to the clinic, we could be talking about making joint replacements obsolete.
It's a huge shift. This approach doesn't just manage symptoms. It targets and potentially reverses the fundamental cause of the disease.
So to really get this, we have to start with the mechanism. The biology that drives this breakdown in the first place.
We do. And it centers on a concept these scientists first identified a couple of years ago. Proteins they call gerozymes.
Gerozymes, I like that name.
Yeah.
It's sort of a mix of gerontology and enzyme. So let's break that down. What are these proteins?
Fundamentally, they are proteins whose activity, it just ramps up as we get older. And they aren't just passive markers of aging. The research shows they are active drivers of decline.
So if their levels go up, our tissue function goes down.
Precisely. They throw the whole cellular environment out of balance. They drive that loss of function, of elasticity, of our body's ability to repair itself. They're like an accelerator pedal for age-related decline.
It sounds like a whole new class of therapeutic targets then. You're not just treating one disease symptom. You're going after the underlying mechanism of aging that makes these diseases possible.
That's the idea. And the specific gerozyme at the heart of this study, the one they targeted in cartilage, is called 15-PGDH.
15-PGDH. I'm assuming that stands for something a bit more complex.
It does, 15-hydroxyprostaglandin dehydrogenase. But we'll just call it 15-PGDH.
Okay, so when the researchers looked at arthritic joints in their models, what did they find with this 15-PGDH?
They found a really clear, really powerful correlation with age. When they compared the knee cartilage of old mice to young mice, the level of this protein had increased by about 2-fold.
Doubled. That's a huge jump.
It's a highly significant spike. It immediately established 15-PGDH as a key molecular signature of cartilage aging, and it strongly suggested this protein was an active player in the whole deterioration process.
So you have this protein that doubles in concentration as the joint breaks down. Now, was this a totally new discovery for this protein, or did they already suspect it was involved in aging?
And that's where the story gets really compelling, because this group had already studied 15-PGDH in other tissues.
So this wasn't its first appearance as a villain, so to speak.
Not at all. And that's why they were so confident in calling it a master regulator of aging.
Let's get that context then. Where else does this protein cause trouble?
Well, their earlier work had shown a huge role for it in muscle health. For instance, if they gave a small molecule drug that blocks 15-PGDH to old animals, those animals saw a significant increase in muscle mass and in their physical endurance.
So it's like a reversal of age-related muscle wasting. Sarcopenia.
It is, yes. And they prove the opposite, too. If you take young mice and you genetically engineer them to overexpress 15-PGDH to flood their systems with it, their muscles weaken and shrink. It's like inducing accelerated aging.
So in muscle, it's a clear switch. High levels mean decline. Blocking it means improvement.
A very clear, unambiguous switch.
And it's not just muscle, right? I saw mentions of other tissues.
Correct. Its role appears to be widespread. Blocking 15-PGH has also been shown to boost regeneration in bone, in nerve tissue, and even in blood cells.
So the evidence was already piling up that this protein acts as a systemic brake on our body's ability to repair itself.
Which provided a very strong reason to test it in cartilage, even though cartilage is, notoriously difficult to fix.
Okay, so to understand how blocking it helps, we need to know what it's actually doing. What is the molecular lever this gerozyme is pulling?
That lever is its job as an enzyme. The main function of 15-PGDH is to degrade, to destroy, a really important signaling molecule. That molecule is called prostaglandin E2, or PGE2 for short.
The prostaglandin E2 connection. Now, that's a molecule that I think a lot of people have heard of, often in the context of inflammation.
It has a bit of a double-edged reputation, that's true.
So why is getting rid of it a bad thing here?
Because when it's kept at the right biological level, prostaglandin E2 is absolutely essential for regeneration. Previous research had already shown it's critical for muscle stem cell function. It's like a go signal for healing and growth.
So prostaglandin E2 is the foreman on the construction site telling the cells to start rebuilding.
That's a great way to put it. And 15-PGDH is the molecular vandal that's constantly running around tearing up the foreman's blueprints.
I see. So as we age, we get twice as many of these blueprint shredders in the joint.
Exactly. Which means any regenerative signal from prostaglandin E2 gets destroyed almost immediately. The go signal is constantly being silenced by this overactive enzyme.
And that leads us right to the core hypothesis they decided to test.
Yes. If aging increases the blueprint shredder, 15-PGDH, which in turn destroys the repair signal, prostaglandin E2,
then blocking the shredder should allow the repair signal to build back up, and maybe, just maybe, that would allow the cartilage to finally start regenerating itself.
That was a hypothesis, an elegant one, but it was up against a huge biological challenge because of the tissue they chose.
Articular cartilage. Before we get to the results, let's just emphasize why this tissue is often considered a biological dead end.
Right. And to be specific, we're talking about hyaline cartilage. This is that smooth, glossy, super low friction surface on the ends of your bones in the big joints like your knees and hips. It's what allows for painless movement.
And it has no direct blood supply, which is a major problem for healing.
A huge problem. That lack of blood flow, plus the incredible mechanical stress it's under, means it has almost zero capacity to regenerate on its own. It can't heal like bone or muscle can.
And the search for stem cells in cartilage hasn't been very successful either.
It hasn't. Decades of searching for a reliable population of tissue-specific stem cells have mostly come up empty. That's why joint degeneration has always been seen as irreversible. And that's why doing this with a drug is so also groundbreaking.
Okay, let's get into it. Experiment 1, reversing existing age-related damage in mice. What was the setup?
They took aged mice whose knee cartilage was already showing clear signs of being old and worn out. It was thinner, it was weaker.
And the treatment?
They gave them a small molecule drug designed to be a potent inhibitor of 15-PGDH. And they tested it 2 ways, which was smart.
What were the 2 delivery methods?
First was a systemic injection into the abdomen, so the drug would circulate through the whole body. Second was a local injection, delivering the inhibitor right into the knee joint itself.
And what happened to that old, thin cartilage?
The results were remarkable. In both the systemic and the local treatment groups, the cartilage that had been so thin and degraded, it actually thickened significantly across the whole surface of the joint.
But the big question is always what kind of cartilage grew back. The body can sometimes make a sort of scar tissue, fibrocartilage, which isn't as good.
And that's the crucial point. They verified it. The new tissue was functional hyaline cartilage. The good stuff. It wasn't just a scar, it was a proper, high-quality biological repair.
That must have been a surprise, even for the researchers.
It seems so. Dr. Bhutani, one of the senior authors, said, and I think this quote says it all, "Cartilage regeneration to such an extent in aged mice took us by surprise. The effect was remarkable."
When the experts doing the work are surprised, you know you're onto something big.
It suggested they'd found a really fundamental regenerative switch.
Okay, that covers reversing existing damage.
Well,
But experiment 2 shifted focus to prevention. which could have a huge impact for younger people.
A massive unmet need, yes. Think about sports injuries, specifically something like an ACL tear, which is so common.
Right. Even if you get it surgically repaired, your risk of arthritis in that knee later on is much higher.
It's about 50%. 50% of people who tear their ACL will develop pretty bad osteoarthritis within 10 to 15 years. The injury starts a ticking clock on that joint.
So how did the inhibitor work in this injury model?
They induced an injury in mice that mimics an ACL tear, and then they gave a series of injections of the inhibitor twice a week for 4 weeks. They were trying to stop that destructive process right after the trauma.
And what happened in the mice that got the injury but not the drug? The control group.
In the untreated animals, the injury itself caused the 15 pGDH levels in the joint to double almost immediately, so the trauma triggers the aging regulator, and predictably that led to severe osteoarthritis developing very quickly.
And in the animals that got the treatment.
The treatment was incredibly effective. It dramatically reduced the chances of osteoarthritis developing. But they didn't just look at the tissue, they looked at function.
How do they measure that?
They monitored how the animals moved, how much weight they put on their legs. The treated mice moved much more normally. They didn't limp or favor the injured leg. They were putting more weight on it, which shows the biological repair was actually reducing pain and restoring function.
And that's the goal, a repair that improves quality of life.
That's the only goal that matters, really.
Okay, we've got old mice, we've got injured mice, but now we have to get to the most important part. Human relevance. Experiment 3.
This is the essential bridge. They got cartilage tissue from patients who were undergoing total knee replacements.
So you have to understand what this means. This is not just slightly damaged cartilage. This is end-stage, severely degraded tissue.
This is tissue that was being thrown away, taken from people with the worst form of the disease. It's the biological equivalent of a burnt-out engine.
If the inhibitor can do anything with this tissue, that's a paradigm shift. What do?
They took these human cartilage samples, cultured them in a lab dish, and treated them with the 15 pGDH inhibitor. And for a surprisingly short time, just 1 week.
1 week. And what happened?
Even this severely degraded human tissue, it started to show a regenerative response. The number of cells expressing that bad aging regulator went down, the genes for tissue degradation went down, and most importantly, the sample started to actively regenerate a new articular cartilage matrix.
To see any regeneration in tissue that was literally destined for medical waste. It's incredible.
It really is. It suggests that even in the worst case scenarios, the cellular machinery for repair is still there. It's not gone forever. It's just being suppressed.
And this drug seems to lift that suppression.
It flips the switch back to on.
Now, this brings us to the mechanism, because this is where the science gets really, really interesting and challenges what we thought we knew.
It does. Based on what they'd seen in muscle and bone, the logical assumption was that this regeneration must involve stem cells.
Right. You'd expect new worker cells to be coming in to do the rebuilding.
That's the classic model. In most regenerative situations, you need those tissue-specific stem cells to multiply and then turn into the new cells you need. But that's not what happened in cartilage.
They found no stem cells were involved. How are they so sure?
They just couldn't find the evidence for it. There was no sign of stem cell proliferation or differentiation that could account for the amount of new cartilage they were seeing. Dr. Helen Blau, another of the senior authors, was very clear about it. She said, "We were looking for stem cells, but they are clearly not involved." It's very exciting.
So if it wasn't new cells, what was it? Who was doing the rebuilding?
It was the existing cells, a process we call cellular reprogramming. The mature cartilage cells, the chondrocytes that were already there, they simply changed their behavior.
They changed their gene expression.
Exactly. They basically adopted a more youthful, more regenerative identity. It's like they de-aged themselves and went back to an earlier stage where their job was to build, not just maintain.
So you don't need to bring in new construction workers. You just have to convince the tired old crew to pick up a new set of blueprints and get back to work.
That's a perfect analogy. And they could actually track this shift by looking at the gene profiles of the cells before and after the treatment.
Let's break down those numbers, because this really proves the identity change is happening.
Okay, so you can think of it in 3 key populations of cells. First, the bad guys. The cells expressing high levels of 15 pGDH and degradation genes. That population fell from about 8% of the total down to just 3%.
So a huge reduction in the cells causing the problem.
A massive reduction. Second, the cells that make the wrong kind of repair tissue, that scar-like fiber cartilage. That population also dropped from 16% down to 8%. It was cut in half.
So the treatment was steering the cells away from a bad repair.
And pushing them toward a good one, because this is the most important number. The population of cells expressing the genes for high-quality hyaline cartilage formation.
The ones you actually want.
The ones you absolutely want. That population nearly doubled. It surged from 22% of the cells before treatment to an amazing 42% after.
Wow. So you're simultaneously silencing the destructive cells and doubling the number of regenerative cells.
It confirms that there's this large existing pool of cells in our cartilage that has this latent ability to regenerate. It's just locked away. And inhibiting this one aging regulator is the key that unlocks it.
Well, let's circle back to prostaglandin E2 for a second, because there's a potential confusion there for people. We associate it with inflammation, with pain. You take anti-inflammatory drugs to block prostaglandins.
A critical point. And you're absolutely right. High levels of prostaglandin E2 are generated during an injury, and those high levels are what cause pain and can contribute to tissue breakdown.
So why is boosting it a good thing here? It sounds contradictory.
It's all about the dose. The researchers clarified that this molecule works on a spectrum. What this study showed is that at normal physiological levels, a small controlled increase is actually anabolic. It's pro-growth.
So a small increase equals repair, but a huge inflammatory spike equals pain and damage.
Precisely. They found the therapeutic sweet spot. By blocking 15-PGDH, they're not flooding the joint with prostaglandin E2. They're just preventing it from being constantly destroyed. This raises a level just enough to trigger that regenerative gene shift without pushing it into the high inflammatory zone.
It's like turning a dimmer switch up just a little, not flicking on a floodlight.
Exactly. They are restoring a subtle but critical signal that the aging process had silenced.
Which brings us to the implications, what this could all mean. We're talking about a drug, maybe a pill or a simple injection that could regenerate cartilage.
The potential benefit for patients is just enormous. For millions of people who are facing years of pain or a major surgery, this could be a third option, a non-surgical biological restoration of their own joint.
And the mechanism is so powerful because it's using the cells that are already there.
That's what Dr. Bhutani highlighted. He said, by targeting these cells for regeneration, we may have an opportunity to have a bigger overall impact clinically. It's an activation approach, not a replacement approach.
And what's so exciting is that the drug itself isn't some far-off fantasy.
No, and this is maybe the most surprising part of the whole story in terms of the clinical timeline.
So where do things stand with a 15-PGDH inhibitor drug right now?
This is the really good news. An oral version of a 15-PGDH inhibitor is already in phase 1 clinical trials as we speak.
But not for cartilage, right? The trial was started for something else.
That's right. It was launched with the primary goal of treating age-related muscle weakness, sarcopenia, which, as we said, is another condition driven by this same master regulator.
But phase 1 trials are all about safety.
Exactly. They test safety and dosing in healthy volunteers. And those trials have already come back with positive safety data. They've shown the inhibitor is, quote, safe and active in healthy volunteers.
That is a massive head start. They've cleared the first and often the longest hurdle in drug development.
It's a huge shortcut. The path is now clear to design new trials to test this exact same drug specifically for its effects on joints and osteoarthritis.
And the researchers seem optimistic that will happen soon.
Dr. Blau expressed that hope, yes, that a trial focused on cartilage regeneration will be launched soon. So the move from the lab to the clinic for this specific use could be much, much faster than usual.
So to synthesize this for everyone listening, the core takeaway is just profound. By blocking a single master aging regulator, 15-PGDH, scientists were able to trigger cellular reprogramming.
And that reprogramming told existing mature cartilage cells to become young and regenerative again. And critically, this was all done without stem cells. It proves the potential for repair is already in the tissue, just waiting to be unlocked.
And if you could do this for cartilage, and we know it also works for muscle, nerve, and bone, it really makes you rethink our whole approach to age-related disease.
It does. It forces you to consider the bigger picture. Instead of treating arthritis over here and muscle wasting over there, what if you could target the fundamental process of aging that drives all of them?
It could be the most powerful strategy for extending not just our lifespan, but our health span. The years we live well.
The potential here isn't just about avoiding a knee replacement. It's about the possibility of reversing aspects of cellular aging throughout the body just by flipping one molecular switch.
A powerful thought to end on. We'll all be keeping a very close eye on the news about 15 PGDH inhibitors in clinical trials.
The future of mobility and regeneration might be getting here a lot sooner than we thought.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.